Today we are discussing the topic interaction of cells with the extracellular matrix, integrins, focal adhesion and hemidesmosomes. First of all, we discuss the topic extracellular matrix. In biology, the extracellular matrix, also known as ECM, is a collection of extracellular molecules secreted by cells that provide structural and biochemical support to the surrounding cells. And ECM is a collection of extracellular molecules and it is secreted by the cells and it provides the structural and biochemical support to the cell. And the extracellular matrix ECM is the non-cellular component. It is a non-cellular component present within all tissues and organs. The ECM is seen in all tissues and organs and it provides not only essential physical scaffolding for the cellular constituents but also initiates crucial biochemical and biomechanical cues. And the ECM is providing the essential physical scaffoldings to the cells for the cellular constituents, and also it initiates crucial biochemical and biomechanical cues and that are required for tissue morphogenesis, differentiation and homeostasis. The ECM is a complex arrangement of molecules filling in spaces between the cells. ECM is the complex arrangement of molecules that is filling in between the cells, and it is not an amorphous jelly or glue but highly organized structure mostly found in connective tissue. And ECM is mostly found in connective tissue such as tendon, cartilage, bone, dermis of skin, etc. Diverse structures created by different amounts and organization of ECM components, and ECM components has diverse structures and it has different functions. ECM is a local product for local cells. Cells secrete ECM that is finally assembled outside the cell. ECM is a local product. The local cells produce ECM for their own cells. It is not transported from other sites. And here is the picture of extracellular matrix. It is a skin, epithelium, it is base membrane and inner cells. These are assembled to form extracellular matrix. It provides the structural it provides the strength for the cells. And then we are discussing some functions of extracellular matrix. Due to its diverse nature and composition, the ECM can serve many functions such as providing support. The ECM provides support for the cells and segregating tissues from one another and regulating intracellular communication. The tissue segregation is done by the extracellular matrix and regulating intercellular communication. The information from between the cells is regulated by the extracellular matrix. The extracellular matrix regulates cells' dynamic behavior. ECM regulates the dynamic behavior of cells, which means the cell division and other biomechanical processes is regulated by the extracellular matrix. Then next we are discussing the topic integrins. Integrins are transmembrane cell signaling proteins. They are cell signaling proteins, which means the integrins transport signals from outside to the cell and from inside to outside. Integrins are transmembrane receptors that facilitate cell to cell and cell to extracellular matrix adhesion. It is a transmembrane receptor and it provides the information between cell to cell and cell to extracellular matrix. Upon ligand binding, the transmembrane receptor is provide the adhesion between cell to cell and cell to extracellular matrix. Upon ligand binding, integrins activate signals transduction. The integrins is the center for signal transduction between cell to outside and cell to cell. It activates cell transduction pathways that mediates cellular signals such as regulation of cell cycle, organization of intracellular cytoskeleton, and movement of new receptors to cell membrane. And the information is transferred for these processes through integrin proteins. Integrins. The presence of integrins allows rapid and flexible responses to events at cell surface. Example, signal platelets to initiate an interaction with coagulation factors. The presence of integrins is initiating the rapid response at the cell surface. For example, in coagulation of blood, platelets is trapped in the fibrin network and it is initiated by the integrins at the site of the wound. Then next we are discussing the structure of the integrins. Integrins are obligate heterodimers composed of alpha and beta subunits, and integrins is made of alpha and beta subunits. Integrins are assembled from 18 alpha and 8 beta subunits in man. In Drosophila, 5 alpha and 2 beta subunits. And in Caenorhabditis nematodes, 2 alpha subunits and 1 beta subunit. The alpha and beta subunits are both class 1 transmembrane proteins, so each penetrates the plasma membrane and once can possess several cytoplasmic domains. And the alpha beta subunits are class 1 transmembrane, so it can penetrate in the transmembrane. Suppose this is the transmembrane and the integrins can penetrate through the plasma membrane. This is a picture of integrins and it has a alpha subunit and beta subunit. This is plasma membrane. It is penetrates to the plasma membrane. It provides information from outside to inside and also from the cell to extracellular matrix. Integrins can transport signals in two ways: outside in transport, inside out transport. When one molecule reaches in the end of integrin, when one molecule reaches in the end of integrin proteins, a conformational changes occurs at the 2 alpha and beta chains, segregates and forms a space between them. Through the gap, signaling proteins transport in or out of the cell. When a molecule, suppose this is the plasma membrane and this is alpha subunit, this is beta subunits. When a molecule carrying signal comes outside of the cell, the integrin activates and the alpha and beta subunits segregates each other like this, and the segregation is like 80 angstrom and the signal is transferred into the cell. This is how the information is passed from outside to inside through integrins. The process is same for the transformation of information from the cell to the outside. Then we are moving to next topic. It is focal adhesion. In cell biology, focal adhesion, also known as cell matrix adhesion or FAS, are large macromolecular assemblies. They are large macromolecular assemblies through which mechanical force and regulatory signals are transmitted between the extracellular matrix and interacting cells. Through these focal adhesions, regulatory signals are transmitted between the cells and the extracellular matrix, and it also provide the mechanical force. More precisely, focal adhesion are subcellular structures that mediates the regulatory effects, that is signaling events of a cell in response to ECM adhesion. Focal adhesions are subcellular structures. These are subcellular structures that mediates the regulatory effects, that is signaling events. Signaling events is regulated by the focal adhesion. Also it provides adhesion and mechanical force. Focal adhesion serve as the mechanical linkage to the ECM and as a biochemical signaling hub to concentrate the direct numerous signaling proteins at the site of integrin binding and clustering. Focal adhesion serves as the mechanical linkage to the ECM. The focal adhesion is serve as the strength or the mechanical linkage to the extracellular matrix from a cell, and also it is act as a biochemical signaling hub to concentrate and direct numerous signaling proteins at sites of integrin binding and clustering. The signal transfer through integrin and it is regulated by the focal adhesions. During cell migration and spreading, focal adhesion serve as holding points that suppress membrane contraction and promote protrusion at the leading edge. The focal adhesion is serve as holding points for the cells to prevent the membrane contraction during cell migration, and in stationary cells they serve as the anchorage devices that maintain the cell morphology. For the stationary cells, the focal adhesion maintain their cell morphology, and during cell migration or spreading, it hold the point that suppress membrane contraction. Membrane contraction prevent the focal adhesion is preventing the membrane contraction during cell migration or spreading, and in stationary cells it maintain the cell morphology. This is a picture of focal adhesion. This is cell membrane, this is integrin proteins, and this is extracellular matrix, and these integrins is connected to the extracellular matrix, and these subcellular molecules are known as focal adhesion. This whole unit is known as focal adhesion plaque. Then we are going to next topic, hemidesmosomes. Hemidesmosomes are specialized junctional complexes. They are specialized junctional complexes that contribute to the attachment of epithelial basement membrane in stratified and other complex epithelia. These are the complexes that seen in junctions, and it contribute to the attachment of epithelial basement membrane. The cell is attached to the basement membrane through some structures which is known as hemidesmosomes. It connects the cell to the basement membrane. This is picture of hemidesmosomes. Here the cell is connected to the basement membrane through hemidesmosomes. Hemidesmosomes are very small stud or rivet-like structures. They are very small stud or rivet-like structures on the inner basal surface of keratinocytes in the epidermis of skins. It is seen in keratinocytes of epidermis of skin and in the inner basal surface, and it connects to the basal surface. While desmosomes link two cells together, hemidesmosomes attach one cell to the other to the extracellular matrix. We know that the desmosomes links two cells together, but in hemidesmosomes attach one cell to the extracellular matrix. hemidesmosomes attach one cell to its base or extracellular matrix. Hemidesmosomes use integrins cells as addition proteins. Hemidesmosomes is connected to the basement membrane and it connected to cell through integrins. For example, here is the basement membrane and here is the cell, and through cell membrane we can see integrins, and hemidesmosomes is connected through the integrins and it connects to basal membrane. And hemidesmosomes are asymmetrical. They are asymmetrical and are found in epithelial cells connecting the basal phase to other cells. They are seen in epithelial cells. Their function is maintenance of tissue structure. They are maintaining the tissue structure and anchorage to the basal membrane. Here is the picture of keratinocytes. Here this is extracellular matrix or basal lamina. The integrins, these are integrins, and here we can see the hemidesmosome laminin, and they are connecting the cell.